In this video lecture, we will talk about transcription and we will focus on the mechanism. So I'm going to give you a quick introduction, then I will move to the different stages: initiation, elongation, and termination, and then I will talk a little bit about the regulation. So let's start. So as we talked before, in laboratory experiments, genes can be transcribed and translated after being transplanted from one species into another one. Here, for example, we have a firefly, and we take this fluorescent gene and put it in a tobacco plant. And here you can see how they are expressing all these genes. Now, you can also not only do that with plant, but also you can do it, for example, with pigs. And they are, if you see the nose and here, these structures, the pig is expressing a jellyfish gene. And why we do that? Because, as we said, there is a universal genetic code. Now I want that you watch this video clip of your textbook that introduces the concept of transcription.
The first step in protein synthesis is transcription of RNA from DNA. The portion of the DNA that is transcribed into an RNA molecule is called a transcription unit. An enzyme called RNA polymerase carries out transcription. With the help of protein transcription factors, it attaches to the beginning of a region of the DNA called the promoter, pries the DNA strands apart, and untwists a short portion of the double helix. RNA polymerase moves along the DNA, pairing up RNA nucleotides with their DNA complements, adding nucleotides to the end of the growing RNA molecule. Here's a close-up view of elongation. Only one DNA strand, the template strand, serves as a template for RNA synthesis. RNA polymerase moves along the DNA strand in the 3' to 5' direction, adding nucleotides to the 3' end of the RNA chain. Note that U in RNA pairs with A in DNA.
Once transcription is complete, RNA polymerase releases the completed RNA and detaches from the DNA.
Now let's move to these three stages: initiation, elongation, and termination. To explain the synthesis of the RNA transcript, I'm going to show you very briefly in the following slides these three processes of the initiation, elongation, and termination. And then I will show you a video of your textbook to make sure that you understand the concept. In the initiation, you have the DNA molecule, and this one is separated. Here you see the transcript unit. This is the section of the DNA that it will produce the messenger RNA. And before that, you have a sequence of DNA that is not going to be translated, that is called the promoter, that is going to be in charge of the regulation of the transcription. An RNA polymerase is going to locate in this region, and it will determine the start point of the transcription. Later on, as the DNA unwinds, the RNA transcript will start synthesizing RNA, and this RNA transcript, it will start growing. So in the beginning, as I said, the RNA polymerase will bind to the promoter, then the DNA will unwind, as I show you here, and finally the RNA polymerase initiates the synthesis of the transcript. Then it will be the elongation, and as you can see here, the elongation will go always from 5' to 3' in the transcript. And this is the direction, you can see it here, of the newly made RNA that come from a template strand of DNA. So as I said before, the polymerase moves downstream of the unwinding DNA, and it always elongates the RNA transcript from a 5' to a 3', and later, in the wake of the transcription, the DNA strands will reform. And as you can see here, the RNA polymerase will be released, and the complete transcript will be also released to be processed as is described in your textbook. So eventually the RNA transcript will be released and the DNA polymerase will be attached from the DNA. Okay, to finish this section on transcription, we already discussed a little bit about the introduction of the transcription. I told you that there are three main phases that are initiation, elongation, and termination. You watched several videos. Now we will talk about how transcription is regulated. To talk about the regulation of transcription, we need to talk about the data box, and we have to talk about promoter. And a promoter is going to be the DNA sequence where the RNA polymerase is going to bind to start the initiation of transcription. A promoter is a sequence of DNA that in this graph it shows two main regions. One that is the data box, that is a highly conserved sequence of DNA, and also have a starting point for the transcription. So the promoter is this DNA sequence that in this case, if you see it, is upstream of the gene that is going to be transcribed. And this promoter is going to signal the initiation of the transcription. So I told you first that the promoter signals the initiation of the RNA synthesis. And I also told you that a promoter is a region of DNA located near the transcription start sites of genes. So here you can find the start point, and here you can find the promoter. Now we're going to talk about transcription factors. And transcription factors are very important proteins because they control the rate of transcription. In other words, how much of the information, genetic information of the DNA is going to move into messenger RNA. And how they do it, if you remember, this green region is called promoter, and remember that a promoter will signal the initiation of the transcription. So the RNA polymerase is going to go there and is going to attach. And the transcription factors, they will help the RNA polymerase, they will help this protein to find the promoter sequence. And how they do it? Because they help to find the binding site. So again, there are just a lot of physical interactions here, and how the promoter region and the RNA will interact just by binding each other, and the transcription factors will help to do that. So let's recap. I told you the first thing that I want you to remember is that there are proteins that are very important that are called transcription factors, and that they control the rate of transcription. In other words, how much of the DNA will be transformed into messenger RNA. So if you have more of messenger RNA, if you need a lot of transcript, they will control that. If they need less, they will also control that. And what is the mechanism? And we will study this more in detail in genetics. But what they do is they help eukaryotic RNA polymerase to recognize the promoter. So I show you that here you have your RNA polymerase and it's going to bind in the promoter, and they help the RNA polymerase to identify this green region that you see in this diagram. And how they do it? Because they mediate the binding of the RNA polymerase into the initiation factor. And I told you that there were many physical interactions in the case of the RNA and the promoter, and these proteins helped that they attach.